Hello everyone, you are watching sqadia.com and today our lecture topic is spinal cord and we are on the section 3 of this lecture topic, that is the classification of nerve fibers. In this, we will be going through how the nerve fibers are actually classified, what are different types of nerve fibers, what are A, B and C types, why they are called such types, and what are the different subtypes of type A fibers, that is alpha, beta, gamma, delta fibers, and why they are called alpha type fibers. How the myelination, that is how the myelin sheath is actually woven around the axons of the nerves of the peripheral nervous system, how this process of myelination occurs, and what are the nodes of Ranvier and what is the solitary conduction system. Coming to the classification of nerve fibers, there are basically three types: the A type, the B type, and the C type. A type is further subdivided into alpha, beta, gamma, and delta type. A alpha type are actually those are the highly myelinated and have the highest conduction of velocity. Actually, these are divided on the basis of conduction of velocity. Those can conduct at a very high speed are called as alpha type or A type because they have a lot of myelination. While for example, C type fibers are actually non-myelinated and their conduction velocity is much less as compared to the A type fibers. This is the graph which is actually comparing alpha A type fibers and giving us if you see the conduction velocity is almost of alpha type A type fibers is 70 meter per second, and then it is beta is 40, gamma is 10, and delta is 6 meter per second. If you compare that with the fiber diameter, A type fibers are thick. Why? Because they're thick because there are a lot of myelination across going on them. Their size is almost 20 microamperes, beta is 5, gamma is 3, and delta is 2. What are the major function is the motor skeletal muscle. These are the classical alpha A fibers are present in the typical reflex of motor skeletal muscles. Betas are involved in the sensory, touch, pressure, and vibration, mainly the dorsal column tract. Muscle spindles actually carry the gamma type fibers because they have to have a conduction velocity which is relatively slower compared to the A type fibers. While delta type, they are mainly pain, sharp, or localized temperature tough. These fibers are mainly present in the spinothalamic tract, which is actually responsible for carrying out the sensation from via anterolateral system in the spinal cord. Coming to the B type and C type fibers, if you just compare it, the B type and C type fibers conduction velocity to the alpha type was 70 meter per second, but on the other hand B type are just 3 meter per second and C types are 0.5 meter per second. And as you can appreciate over here is that only C type fibers are non-myelinated. Almost all others are myelinated, but actually it is the number of swirls of the Schwann cells around a single axon. If there is a lot of swirls of Schwann cells around a single axon, this means it would be highly myelinated. That will also not only increase the diameter of the axon, but that would mean that it will increase the conduction velocity of them as well. And if you can appreciate, these are actually B type fibers are present in a preganglionic autonomic areas, while the C type actually diffuse pain, temperature, and postganglionic autonomic. The free nerve endings which are present as in our hands and in the skin are actually from the C type fibers. These are non-myelinated fibers, but they carry the sensation and of pain and temperature. While the other sensation which is carried by the pain temperature is actually through the spinothalamic tract, which is basically delta type A type fibers. Now coming to the myelination in peripheral nervous system. The myelination done by the peripheral nervous myelination is done by the Schwann cells in the peripheral nervous system. While in case of it was the oligodendrocytes in the CNS, it is the Schwann cells here in the peripheral nervous system. If you can appreciate, this table is actually showing a very different concept. In another lecture, I told you that a single oligodendrocytes actually surrounds almost up to 50 to 60 of these nerve fibers in the central nervous system or in the brain. But over here, instead, in the central nervous system, when you go to the peripheral nervous system, it is a single Schwann cell is actually covering the single axon, that is a single nerve cell. So it's 1 to 1 ratio, but in the central nervous system, it is 1 to 60 ratio. Nodes of Ranvier are present in the both. There are incisors, Schmidt Lantman incisors are present in the both. Max axon is present in the nervous system but not in the CNS. Now this is an example of this is a single axon, and actually Schwann cells initially start surrounding it, and then it is actually swirling around. It's woven. It's like it's weaving around this axon and keeps on rotating and rotating, and eventually, as you can appreciate, not only there is number of sheets are increasing, similarly the diameter is also increasing. So for example, in type A alpha fibers, there is a lot of actually myelin is present, so they have a large diameter. Similarly, when the lot of myelination is already there, they have a very high large conduction of velocity compared to other fibers. Now coming to the what is nodes of Ranvier. Now this is a cut section of an axon which has actually already been myelinated. As you can appreciate, this is the axon in the center. If axon is cut like this is where you can appreciate, and then this is the Schwann cells which have actually surrounded this axon. But where one Schwann cells end, another Schwann cell starts over here. Now there's a gap at this point. This is termed as the nodes of Ranvier. If you see it on the lateral view, actually you appreciate that there is high grade of myelination. Then is a very small area, maybe in microns, which is not myelinated, and then again the myelination starts. Now this is the point, this is the area, this is termed as the nodes of Ranvier. What is the role of nodes of Ranvier is that actually they are helpful in saltatory conduction. When we say saltatory, means that nerve impulses do not travel along a straight line. Rather, they jump from one nodes of Ranvier to another nodes of Ranvier, then to another, and then to another. So this jumping of nerve impulses from one node to another node is actually called as saltatory conduction. Why? Because all other area is actually insulated. Now the myelination not only helps in insulation but also helps in insulation of the axon from the electrical impulses. So if electrical impulse has to travel along the axon, it has to travel from one node of Ranvier to another node, and then it will jump to the third node and keep on jumping from node to node, eventually reaching the effector organ. So this is not only results in a saltatory conduction, but actually this also you can appreciate that actually myelination is helpful in insulation of axons from other stimuli, similarly protecting it from injury. So in this section we have covered what the myelination occurs in the peripheral nervous system, what is the classification of the nerve fibers, why they are classified as A type, B type, and C type, and what is the role of myelination, how the Schwann cells actually myelinate a single axon in the peripheral nervous system, and what is the nodes of Ranvier and what are their role in saltatory conduction of nerve impulses across the axon. For more information, keep watching sqadia.com. Thank you very much.